scientists have this really cool way to kind of trick these tiny microbes into making lots and lots of valuable stuff.
Yeah, like we're talking about coaxing them into becoming super producers.
Right. And it all comes down to this technique called analog resistant selection.
Analogs, you can think of them like imposters, molecules that look like the real deal, enough to, throw off the microbe's internal workings.
So it's like we're using these imposters to mess with their system and get them to overproduce things we need.
Exactly. And what I find so fascinating is that this whole approach, it's like fine-tuning a microbe's metabolism, but to our advantage.
Yeah, kind of like hacking into their system for good.
Yeah.
So in this deep dive, we're going to explore how scientists use this analog resistance to get microbial strains to produce these essential building blocks. What are those called again?
Primary metabolite.
Primary metabolites. And we've got some really good examples from our sources to illustrate this whole process.
Absolutely.
All right, so let's kick things off with L-lysine overproduction in Corynebacterium glutamicum. Now, L-lysine, that's a pretty important amino acid, right? And scientists figured out that by exposing C-glutamicum to this compound called diaminoethylcysteine,
Or DAEC for short.
DAC, right, they could actually isolate strains that were resistant to it.
And the big question is, why were these strains resistant, right? And often it boils down to mutations in a specific gene.
Which one?
It's called the ASC gene. And what this gene does is it provides the instructions for building an enzyme called aspartakinase.
Aspartakinase, got it.
And this enzyme, it's a key player in lysine production. Think of it as like a thermostat, for controlling the process.
Okay, so aspartakinase is the thermostat controlling how much lysine gets made.
Exactly. Now, normally when there's enough lysine around and another amino acid called threonine, they basically tell aspartakinase, hey, slow down production. It's like a feedback mechanism.
So lysine and threonine act like signals saying, we're good, you can chill now.
Precisely. But here's where it gets interesting. In these DAC-resistant mutants, the aspartakinase, it's mutated.
So it's different.
Yeah, and because it's different, it's less sensitive to those slowdown signals from not only lysine and threonine, but also DAEC itself.
So it's like the thermostat is broken.
In a way, yeah. And as a result, these mutant strains, they keep churning out lysine even when there's plenty of it.
Which leads to that overproduction we were talking about?
Right. And a good example of such a strain is Corynebacterium glutamicum strain, AG3456.
AG3456, all right, got it. So this isn't just some lab experiment, it's having a real impact on how we make essential nutrients, right?
Absolutely. It's being used in the real world.
Okay, let's move on to another example. How about L-threonine production in Escherichia coli?
Okay, so with L-threonine, it's a similar strategy, but the analog we use here is alpha-methylthreonine.
Alpha-methylthreonine, okay.
And just like before, we isolate E. coli strains that are resistant to this analog.
And I bet there's a specific gene involved here, too.
You bet there is. And if you look closely, we'll see a pattern emerging. Resistance to alpha-methylthreonine often comes from mutations in the THR gene.
THR, all right. What does that one do?
Well, THR, it encodes another key regulatory enzyme.
Another one of those control points.
Yeah. And this one's called aspartakinase, ihomoserine dehydrogenase.
Wow, that's a mouthful.
It is. But this enzyme basically governs an early step in threonine production.
Okay, so it's like the first checkpoint in the assembly line.
Exactly. And guess what? Just like with lysine, these mutations in threon A make the enzyme less sensitive to the feedback inhibition from threon A.
So once again, it's messing with the brakes on production.
Right. The brakes are weakened, and the E. coli strain becomes a more efficient threonine factory, like the E. coli strain ATCC21277.
That's ATCC21277.
Okay, so we got lysine, we got threonine. What's next?
Let's talk about L-tryptophan overproduction in Bacillus subtophan.
All right, bring on the tryptophan.
In this case, the analog we're using is 5-methyltryptophan.
5-methyltryptophan, got it.
And what's interesting here is that there are actually a couple of ways this resistance and subsequent overproduction can happen.
Oh, really? Like different pathways?
Yeah, different ways the microbes can achieve the same outcome. Okay, I'm curious. Tell me more about these different pathways.
So one way involves mutations in a gene called TRPE. Now this gene, it provides the blueprint for an enzyme called anthranilate synthase.
Anthranilate synthase, okay.
And this enzyme, it kicks off the whole process of tryptophan synthesis. It's like the first step of the production line.
So it's crucial.
Very much so.
Yeah.
And a mutation in TRPE can make this enzyme resistant to the feedback inhibition from tryptophan.
So the very first step becomes insensitive to how much tryptophan is already there.
Precisely. Now the second mechanism, it involves a different gene, TRPR.
TRPR. All right, what's special about this one?
Well, TRPR encodes a repressor protein called TRPR. And this protein, it normally acts as a stop sign when tryptophan levels are high.
So it's like the master control switch.
Exactly. It halts the activity of what's called the trap operon, which is basically a group of genes all working together to produce tryptophan.
Like a coordinated team effort.
Yeah, and mutations in TRPR, they can either weaken or totally eliminate this shutdown signal, leading to increased tryptophan production.
So we can either mess with the local control, the enthranolate synthase, or the master control, the TRPR, and both can lead to overproduction.
You got it. And our sources also touch upon nucleotide overproduction.
Oh yeah, nucleotides. Those are the building blocks of DNA and RNA, right?
Exactly. And this just highlights how versatile this analog resistance method is.
So it's not just limited to amino acids?
Not at all.
Oh.
Scientists have used analogs like 8-azogranine, which is a purine analog, to select for microorganisms that overproduce nucleotides, like GMP and IMP.
GMP and IMP, those have lots of uses, right?
Yeah, a wide range of industrial applications. And the underlying principle, again, is to disrupt the feedback control.
By creating these mutants.
Exactly. And these mutations can be in the genes responsible for either making or regulating these nucleotides.
So messing with either the production itself or the control mechanism.
Right. It's all about deregulation, taking the brakes off production.
Okay, so we've seen a bunch of examples now. What's the main takeaway from all of this?
Well, if we look for the common thread running through all these examples, it's the ability of analog resistance selection to help us isolate these mutant strains.
With those altered enzymes.
Exactly. And these alterations, they consistently lead to either a reduction or a complete loss of feedback inhibition or repression.
So they just keep producing even when they shouldn't.
Exactly. They're like little factories that never stop.
And this is huge for biomanufacturing.
Absolutely. It makes the whole process more efficient and economical.
It's amazing how a tiny change at the genetic level can have such a big impact on an industrial scale.
It is. And that brings us to the power of analog resistance selection. It's become a really important tool in the field of metabolic engineering.
We're essentially guiding evolution at the molecular level.
We are. So here's something to think about. How does this targeted approach to manipulating microbes compare to the traditional ways we've been improving production in organisms for centuries, like, breeding and selective cultivation? And what does this mean for the future of bio-based products?
That's something to ponder indeed.
It is.
Lots to think about there.
Yeah.
Thanks for this deep dive.
My pleasure.
It's been really illuminating.
Glad to hear it.